Hello students, now we will learn about different phases of growth in case of plant body during its lifespan. First one, germination phase. And vegetative phase. Second one, reproductive phase. Here we see a formation of flowers. Right? We see flowering. Third phase is senescence and death. First one, germination phase. Or vegetative phase. In some types of plants, we see vegetative propagation. So there we are not giving term like germination, because this is the term that we use for the seeds. Germination, also called as sprouting in seeds. Commonly we say germination, also called as sprouting. So during germination, first step is imbibition happens. Imbibition means initial sticking of water molecule onto the outermost layer of the seed. That is called as adsorption. AD, adsorption. So imbibition means initial or first adsorption of water on seed layer, seed outer layer, and later water enters into the seed, that is called as absorption. Into seed. Because of entry of water molecule into the seed, gibberellic acid content increases. So secondly, gibberellic acid or gibberellin, phytohormone gibberellin, increases in content. Later, gibberellic acid translocates to the alluron layer. Gibberellic acid I am writing as capital G, capital E. Translocates, if it is a monocot, onto alluron layer in monocot or into endospore in dicots. Next, after translocation, we know that alluron layer is a proteinaceous layer. So here we have the food material, stored food material made of proteins. Next. So here we see the proteins. Here gibberellic acid helps in synthesis of proteases and amylases. Acid supports in synthesis and activation of amylases and proteases. Amylases acts on carbohydrates and proteases acts on proteins and breaks them, so that food material will release energy. Next, after releasing of the energy from carbohydrates and proteins. Later, that energy is translocated into radical and plumule. Now, energy in form of ATP translocates into radical and plumule. So when energy has been reached up to radical, then it emerges out of seed. So after emerging of radical from the seed, then plumule starts developing. Because we know that radical is nothing but the root part. Plumule after root formation, plumule develops. So this is about germination phase. Even in vegetative phase also, initially, the radical supports in absorption of nutrients from the soil, means of minerals from the soil and water, and also water also from the soil. Later during vegetative phase completely, it is not up to the reproductive phase. So this is about germination phase. During vegetative phase, just plant keeps on undergoing mitotic divisions and it grows in sets. But during asexual reproduction in some types of plants, vegetative reproduction happens. That we call as vegetative propagation. There is no point of seed formation, but small plantlets will be developed from a few parts of the plant body, like stem, from leaf, from roots, etc. Example, if we take a leaf, bryophyllum is the leaf which is giving rise to new plantlets from its nodes or incision area. So if this is the bryophyllum leaf, we can see formation of new plantlets at this incision means the depressed areas. They are considered as some node-like regions. Next, we will see about types of germination. So, what are the types of germination? First one is epigeal germination. Epigeal type of germination is seen in case of dicots. Here, only hypocotyle elongation is seen. For suppose, if this is the earth layer and if this is the seed, then there are some structures for the seed, like the tip part will be plumule and the bottom part will be radical. Now, this part is called epicotyle and this is called hypocotyle. Here we see in case of dicots, plumule will be along with epicotyle, it will be of same length, but hypocotyle will increase its length and radical will be somewhere here. So, epigeal type of germination is seen in case of dicots. Next one, hypogeal type of germination. It is characteristic feature of monocots, right? Seen in monocots. Here, both epicotyle of the seed and hypocotyle will be elongated. But in case of monocots, we use a term like coleoptile and coleorhiza. Both coleoptile and coleorhiza are elongated. For suppose if this is the earth layer and this is a monocot seed. For example, think that it is a maize seed. So here, so this will be plumule part, and this tip will be coleoptile, and this will be radical part, and this will be coleorhiza. The next stage, seed will be like this, both. Coleoptile and coleorhiza will be elongated. So this will be coleoptile and this will be coleorhiza. Here it will be radical and this will be plume. So these are two types of germinations. One is epigeal and hypogeal type of germinations.